Hey everyone, so in this video we're going to be talking about SCID and other associated disorders, and we're going to go ahead and start with this chart that I made, this flow diagram, and it kind of goes hand in hand with the blood cell differentiation, where we come from a common lymphoid progenitor cell and we're going to end up with natural killer cells, T cells, and B cells. But just keep in mind that all of these different proteins and genes that I have listed alongside here, it's not exactly how it works as far as cell differentiation, but I think this kind of sums up very nicely where each of these components fit in to that diagram, so you can correctly figure out which kind of immunodeficiency the patient has. So just keep in mind that this is not normally how we would draw out blood cell differentiation. This is just to make this concept a little bit easier for you guys to understand. So the common lymphoid progenitor cell can either go into and become a natural killer cell, it can go and become a T cell, or it can go and become a B cell as shown below. What I really want you guys to pay attention to is where all of these things in red, whether it be a gene or a protein, come into play. So as you can see, this protein called ADA, adenosine deaminase, is involved in the creation of both, or I guess I should say all three, natural killer cells, T cells, and B cells. RAG1 and RAG2, which are proteins that are used in VDJ recombination, are necessary for both the formation of the BCR and TCR. So they are going to be important for both T cell and B cell maturation. And the BTK gene is vital for BCR formation. So B for B cell, right? So BTK, BCR. Without the BTK gene, we would not have B cells because we would not be able to form the BCRs initially. The common gamma chain, which is listed right here and right here, is both necessary for T cell and natural killer cell formation. And then lastly, we have AID and AIR. So AID is a protein that adds point mutations during somatic hypermutation, and without AID, we would not be able to undergo somatic hypermutation or class switch recombination. So without AID, we would not be getting B cells with different immune globulins on the surface. So we would only have B cells with IgM, and that leads to something called hyper Igm syndrome, which we will be talking about in just a second. And then the protein AIR is actually a component of something of cells called MTEC. You have CTEC and you have MTEC. CTEC is in the cortex, MTEC is in the medulla. TEC stands for thymic epithelial cells, and these are involved in, MTEC specifically is involved in negative selection. CTEC would be involved in positive selection. So MTEC expresses this protein called AIR, and what AIR does is it allows for central tolerance. So essentially negative selection in general just gets rid of T cells that have too strong of an affinity to either MHC class 1 or 2 molecules. So if we don't have AIR being produced by MTEC cells, then we won't have central tolerance, and that could potentially lead to some autoimmune issues. Okay, so now that we've looked at all of these things, let's go ahead and look at our chart. So this kind of sums up nicely what we had said on the previous section. The only thing that is not covered on that chart is Degeorge Syndrome. And Degeorge Syndrome is usually a de novo mutation, which means that it's not typically inherited, it's sporadic. And what you really need to know about Degeorge Syndrome is that depending on the severity, and it can have varying severity levels from absent of a thymus to maybe partial thymus to a dysfunctional thymus. So typically when you are going to get questions on this, it's going to have either lower or no T cells at all. Obviously, if they have an absent thymus, they won't have T cells, right? Because T cell maturation happens in the thymus, and so without the thymus, you won't have T cells. Another thing to note is that they usually have unusual facial features, cardiac defects, and because they don't have a thymus, they also don't have, they typically have hypoparathyroidism. If you don't have a parathyroid hormones, obviously you're going to have hypocalcemia, which is an important characteristic of Degeorge syndrome. So SCIDs, this SCID stands for severe combined immunodeficiency, and it can actually result from 3 different kinds of deficiencies, either a deficiency in common gamma chain, ADA, RAG1, or RAG2. So let's go ahead and go back to our diagram really quick. So as I previously mentioned on the chart before, you can get a deficiency in either ADA, RAG1 or RAG2, or common gamma chain. And all 3 of these things can lead to SCID, but they will result in varying levels of natural killer cells, T cells, and B cells. So with the deficiency in common gamma chain, as I previously mentioned, you're going to have absence in natural killer cells and T cells because the common gamma chain is necessary for T cell and natural killer cell formation. In a deficiency of ADA, ADA is involved in the formation of natural killer cells, B cells, and T cells. So obviously, we're going to have a deficiency in all 3 of those. In a deficiency in RAG1 and RAG2, that means we're going to not be able to do VDJ recombination for our B cells and T cells. So we're going to have absent B and T cells, but our natural killer cells aren't involved in that process. They're part of the innate immune system, so they don't go through VDJ recombination. And so we will have present natural killer cells, but absent B cells and T cells. A mutation in our BTK gene, which is involved in the BCR formation, if we have a mutation in that, then obviously we're not going to have any B cells. This condition is actually called agammaglobulinemia, which is abbreviated XLA. And many things can point to XLA versus SCID. Obviously, if they give you a flow cytometry, then you'd be able to point out that you have no B cells, but you have T cells and natural killer cells present, right? Another thing that will help you distinguish between SCIDs and XLA is these things called TREC and CREC. So TREC, if you have a deficiency in TREC, that means you have SCID. If you have a deficiency in CREC, that means you have XLA. And the way I think about it is XLA, which is agammaglobulinemia, is involved in B cell formation, right? SCID can be involved in B cell formation, but really the defining feature of SCID is that you don't have T cells, right? That is a commonality with SCID. That defines SCID is no T cells. You might have B cells, you might have natural killer cells, but you definitely don't have any T cells. And so I think of TREC, it starts with a T, so I think of T cells and I think of SCID. CREC is for B cells, and the things that really have to do with B cells are going to be XLA. Hyper-IgM syndrome does as well have to do with B cells, and we're going to talk about that next. But it will have a normal CREC level, and that's mainly because our B cells are still present. They're just not functional in hyper-IgM syndrome. So the next thing is hyper-IgM syndrome, and that arises in a deficiency of AID. AID is a protein that adds point mutations in somatic hypermutation. And if you don't know what somatic hypermutation is or class switching recombination, I would recommend you go watch that video. So if you cannot undergo somatic hypermutation and class switching, you will not be able to class switch from IgM, right? So as our B cells maturate, they are initially IgM, and that's our mature but naive B cells. And in order to become no longer naive, they will undergo class switch recombination and class switch to IgE, IgA, IgG, etc. So if you don't have AID, you can't add those point mutations, you can't go through somatic hypermutation and class switch recombination, so now you're going to be left with all these B cells with only IgM on the surface. So the defining characteristic of hyper-IgM syndrome is obviously an abundance of IgM, but also lymph node hyperplasia. And this happens because our body, doesn't know that we are deficient in AID. We're not that smart. So essentially what's going to happen is we are going to increase our B cells in order to prepare it for the protein AID that is not actually present. So we're going to keep proliferating these B cells that have IgM on the surface, but we are never going to be able to class switch. And so the lymph node will actually undergo hyperplasia due to all of these B cells proliferating. The last condition is APE-CED, but what this stands for is autoimmune polyendocrinopathy candidiasis ectodermal dystrophy. And while that is a mouthful, I actually think it does a pretty good job of helping you understand what that condition is. So it might behoove you to actually memorize what that stands for, at least to a certain extent. So obviously, from the name, we can probably guess that it's going to be something to do with an autoimmune disease. We can probably guess that it's going to cause candidiasis, and it's polyendocrinopathy, so we can probably guess that it has something to do with the endocrine system, and so adrenal insufficiency would not be an uncommon finding. Now, this condition arises from a deficiency in a protein called AIR. AIR is expressed by MTEC cells, which we've already discussed, and it allows for central tolerance. So essentially, without that protein, we're going to have these T cells that arise, and there's going to be some T cells that have an extremely high affinity to bind to self-antigen, which means we're going to end up with autoimmune diseases. So along with these candidiasis and adrenal insufficiency, there's also many other characteristics of this disease that will arise from it just being an autoimmune problem in general. So keep that in mind. So again, all of this has nothing to do with AIR, so our natural killer cells and B cells will be present. T cells are present, but they are overreactive. I hope this summed up these conditions nicely for you guys. If you have any questions, please feel free to leave a comment. Thank you.